said, École Normale Supérieure in Paris, and I work with and so before I work with oops, it doesn't work. Yeah. Yes, it works. It works. Okay, so yes, I'm really glad to be here for several reasons, for scientific reasons, but also because you have a very, very beautiful city. So I've already visited Elephanta Caves. I've seen a beautiful sunset from Marina Drive, and we've also been to a beautiful concert with Uncle Nadata about Indian music. So it's really science and fun. So thank you very much for all that. And so now back to science. The work that I'm going to present you today is the work that I have been performed with collaborators. So the system in France is a bit different from what you have everywhere else in the world. We do not have PIs that have their own team, but there are several PIs, let's say, in the same group. So Hélène Bertrand is an assistant professor, and Nicolas Delsuc, a CNRS researcher. We also have two systems. You can be a university professor or just do research at CNRS. And so the group now is bigger, but the work that I'm going to present you is just with these two staff members, I would say. And of course, this also involves postdoc and PhD students. And each time they will be involved, I will show their pictures. So I thank them really for all that work. And there's also a collaboration with a researcher from other labs and other, let's say, discipline. So we work with, so will that work? Yes, I have to do that here. So we work with a group of medical doctors from Hôpital Saint-Antoine. So they are gastroenterologists, and they work on IBD. And it happens that, okay. It happens that they are specialized in inflammatory bowel disease and Crohn's disease, and there is a lack of superoxide dismutase in this disease, and this is the protein that we mimic. And so, of course, naturally, we had some interaction, but as I will try to show you, it's also important to do imaging. And we've been working with, I'm not used to this kind of pointer, but I will try to use it. So we've been working with physicists from different synchrotron on argon or Soleil to do some imaging, direct imaging of the complexes that we prepare. And as I will explain, we'll also develop specific probes that can be imaged with multimodalities, included infrared and X-fluorescence. So will that work? Yes. Okay, so I will start with telling you a little bit about the periodic table. Of course, this is the emblem of oh, it's a bit strange here. No, there is something remaining that is not on my screen, but okay. Okay, anyway. So this is the emblem of what we do, emblem of chemistry. And the bold atoms are those that are mainly involved in biological system. I will come back to that later. So I'm interested in inorganic chemistry and biology. And originally, if you look at the way that historically organic chemistry was coined, this term was coined, it was from, it's during the 19th century, and it was from chemistry of organized system, and this is the name organic chemistry. And this indicates that at that period, everybody thought that chemistry of life was mainly based on carbon, and mainly with carbon molecule. And so inorganic chemistry was considered at that time as the chemistry of, well, let's say non-living system or material system or general system, but non-living. And what I'm going to tell you is that, of course, as a bioinorganic chemist, I'm pretty much convinced that there is a lot of metal ions in biology, and they are all very important, and they perform fundamental activities. So what are metals? So metals, they represent the largest the largest part of the periodic table. They are indicated here in gray. And what makes them a specific group is the fact that they are all coming here. Okay, shall I try that? They are all coming from from the mine. So that means they come from the earth, and we have to modify them to obtain the metallic element. They share also common properties such as electric conductivity or thermal conductivity, and these properties, these physicochemical properties at the metal zero redox state are corresponding to electrons that are not very strongly attached to the nucleus, and this is also why they can get into an ionic form. They can be oxidized easily, and you can obtain positive metal ions, positive metal ions from them. So now, so metals are used for different, let's say, they can be used for different type of for their properties in the industry. They were also used in the 19th century as medicine. But as I said, at that time in the 19th century, there was really this idea that they are not part endogenously of the biological system. And the first person that showed that is Jules Rollin. So Jules Rollin is a French chemist, and he was working with Louis Pasteur, and Louis Pasteur worked at ENS. So he was exactly at the place where I'm working now. So this is why I mention it. And what is interesting, and it's related to maybe you've seen Amy Palmer's talk, and what he showed is that it was the first to show that a metal ion was necessarily for the growing of a microorganism, Aspergillus niger, and this is zinc. Zinc is important for that. Iron also, but in relation to what Amy showed us, this is nice to see that. Okay, the first person to show that was this guy. And in fact, his idea was they were doing a lot of applied chemistry, and his idea, starting with microorganisms, was to see what were the elements from the periodic table that were important for crops and for growing of animals and so on. And here you see an old photo, or a photo from an old paper, where you see plants that is growing better when it has zinc supplement than when it has not. Okay, now if we look at the chemistry of life of the periodic table, now that we know that metal ions are important, are they all important? So here I've put in orange the bulk elements, so those that were in bold just previously. So of course our body mainly, quantitatively, is mainly made of hydrogen, carbon, nitrogen, oxygen, sodium, potassium, and so on. But there are some very important elements that are present in our biological system that are indicated here in green. Those that I'm going to speak mainly, those that I will going to speak mainly about are the transition element, and I will mainly speak about manganese that is shown here, and a little bit I will mention very quickly copper, and I will mention also rhenium. Where is it? Oh, it's there. Rhenium, that is not a naturally occurring element, and this is why also we use that element for imaging. If I were giving that talk like maybe 15 years ago, I wouldn't have spoken about, about lanthanides. But it has been discovered at the very beginning of the 2010 decades that early lanthanides that are indicated here are also important for life and essential to the growing of some microorganisms that live in the mold of volcanoes. And in the mold of volcanoes, you have a lot of lanthanides from eruptions. Okay, it's coming from the central, well, the core of the Earth, the lower layers. And it has been shown that there some microorganisms, if you suppress the lanthanides, they don't grow anymore. And this is because they use some of these elements in a specific protein that is necessary to the production of their energy. Why metal cations? Why does nature use metal cations? Well, they provide a wide range of different properties that are strong assets, and this is why nature uses them. First, they show a large variety of structure. They also, because they are stable at the N+ state, they show Lewis acidity, so they can play the role of a Lewis acid. They can also be a charge transporter, and they also perform electron exchange. Some of them are also stable in a paramagnetic state. And this is very important because the oxygen that we use for respiration is also a paramagnetic molecule. It has a spin 1, it's in a triplet state. And to interact with spin 0 molecules, it needs to be activated. Otherwise, there's a spin, a spin restriction that makes that the oxygen reacts very, very slowly with our protein and lipids. If I were to, due to the concentration of the oxygen here that we breathe, thermodynamically, my proteins and the lipids and the lipids and the proteins that I have here should be oxidized. They should burn in a way in the air. But thanks to the fact that the oxygen is in a triplet state, there's a kinetic, it's very, very slow process. And if we want to use the oxygen in the biological context, well, biological system needs to activate to bypass this high energy. And there are several ways to do that, but one is to use a paramagnetic stable ion to coordinate the oxygen. And the last part is that there are dynamic structures, and because of that, they can be at the active sites of metalloprotein, coordinating a substrate, modify the substrate, and then releasing the substrate. So this dynamic is necessary if you want catalysis to occur. So here are some. So I've taken just some pictures of some proteins and active sites of important proteins, so they can perform a large range of different roles, and they are in fact present in every fundamental process of biology. So the oxygen, sorry, I forgot. The oxygen transport, electron transfer, oxidation of water into O2, so catalytic activity in a way, O2 activation with cytochrome P450, coordination of DNA, changing the structure. So this is just for molecular recognition. Here you have superoxide dismutase that we will discuss, that are protein that react with the first element in the reduction cascade of the oxygen. From the oxygen, you have then superoxide, H2O2, and water. And so all of these shapes that I've mentioned previously are used in these different proteins. So a variety of structure. Just if you are a chemist, you know that carbon, you can have tetrahedral at the center of a tetrahedral structure. It can be a plane if you have an alkane, and an alkyne if you have a line. What we have with metal ions is a wide range of very, very different structure. So it has been shown at the end of the 19th century that metal ions can be at the center of an octahedron, but this octahedron can be distorted by many different ways, and all very, very in a continuous way, I would say. So you can have a distorted octahedron, or if you elongate at what position, you can remove one ligand in the plane from here, and then you obtain another trigonal bipyramidal structure. You can remove a ligand, the ligand that is below here in this structure, and you obtain a square pyramid. And then you can also go from tetrahedron, make it flat, going to square planar, and you have also a different structure with more than six ligands. So it's a wide range of different structure. Also, a variety of redox states. This is what you see here. So I've just taken the first row, transition element row here, and you see that there's a lot of different oxidation states that are stable. So I took here the example of vanadium with the these different colors. And what is interesting, and that's different from organic chemistry, if you think about organic chemistry, if you exchange electrons one by one in organic chemistry, you generate a radical, and this radical can be controlled by biological systems. There are biological systems working with radical chemistry, but it's not that easy, okay? You need a very strong way to control that. Normally, in organic chemistry, when we transfer electrons, there are 2 by 2s, okay? This is the way it goes. What is very interesting with metal ions is that you can do monoelectronic exchange, and metal ions are used in biological system to either decouple electron donation. If you donate 2 electrons, you need sometimes to decouple this donation, and metal ions can be used to that. Or they can couple, in contrast, the electron donation to provide, for example, 4 electrons in photosystem 2. And all of these properties are controlled, all of these properties are controlled by the redox potential of these elements here. An example here is cytochrome C. It's an IMO protein that contains a porphyrin with an iron 3, and it can exchange electrons one by one to cytochrome C oxidase that is involved in the respiratory complex. And what is interesting is that it changes color, so you can really monitor that, for example, if you do that in a cuvette. Okay, so globally, if we speak about, if you're an inorganic chemist and you're interested in metals in biology, so bioinorganic, what I would like to say here is that this is what drove me to this discipline, is that it's really a multidisciplinary science. In the congress that we have, so this is what we saw in Calcutta last week. There's a lot of different people doing high technological spectroscopies to people working and growing microorganisms, and we all have things to exchange and to discuss, and this is what I think is very interesting. So, for instance, what I will show you is that we do organic chemistry, so we synthesize, for example, for us, ligands. Then we prepare metal complexes and we study them in UV visible spectroscopy. But we need also to develop biochemical interest for metalloproteins. And so there are some bioorganic chemists that purify these proteins and study them out of any cellular context. And as I will explain you, we do the exact opposite to that trend. I mean, purifying from biological context and studying in cuvette. And what we do is that we prepare these metal complexes and we introduce them in cells, and we try to characterize their activity, their speciation, to analyze them in cells. So analyze both their activity and their nature in the cellular context and their distribution. You can also work at the animal level. You need also to understand physics, because as I will show you, we try to develop molecules to do infrared microscopy, and this required discussion with physicists and understanding a little bit of the physics. And the last part I would say is geochemistry. I've not, for my part, worked in geochemistry, but I think it's a very, very important field, and metal ions, especially to understand, for example, the origin of life, or what happens at the bottom of our oceans with this black fumes, for instance. Metal ions are very important to all of that. And so clearly, by being a bioinorganic chemist, you have to, if you are a bioinorganic chemist, you discuss with all of these disciplines, and this is what I think is very interesting. Okay, so now I will say a little more about what we specifically do. So I've already said a little bit what we are interested in is in a way bridging the gap from the round flask to the cell for inorganic systems. And what we are interested in is to characterize the bioactivity of small molecules, to quantify them, try to the label, the symmetry label, are called EG and T2G. And what happens, so I can explain that if you wish, but the labels. And if I take copper 2 is a D9, and if I fill it here, 1, 2, 3, 4, 5. So here you have two possibilities to put the electron, like that or like that. So we say that here there is a degeneracy. The corresponding term would be it would be an E term. So that means it's doubly degenerated for the orbitals. And it has been shown, I think it was in the 1930 by Jan Teller, that such a system is unstable. So that has been first demonstrated theoretically. And it should vibrate in between two states with elongation, with vibration along of one axis. This is what I'm indicated here. So the light Teller can be dynamic, but at some point, if the system is really more stable when you have removed two of the ligand, then it will stay at that more stable state. And here, this is what you have. And you have 1, 2, 3, 4, 5, 6, 7, 8, 9. And this one is more stable than this one. And so copper 2 and manganese 3, for instance, they show this overstability when there is some distortion. And so, of course, you have less negative charge. So these negative charges are supposed to stabilize the positive charge. But in fact, the stabilization of this one, in a way, is smaller than this, let's say, electronic stabilization. And then for copper 2, with this very specific number of electrons that you have inside that, is more stable at this in the square planar. However, it has been discussed that probably in solution, it is possible that there is a far ligand here that could be the solvent. I don't know what discussions are now, but 10 years ago, people were not quite sure that experimentally we could not see transiently a system that would be with 5 ligands. It has been discussed. So, yeah, so, and?
So, it's okay.
No, but you can ask.
So this is favored in some specific regime of the second ionization energy and the energy ranges between the different levels.
Exactly. And this is really an electronic stabilization that is greater than this electrostatic stabilization in a way. That would be given by an additional delta minus ligand. Is that okay? Because you're right, we should add more ligands, but it happens that in that particular situation with that particular number of electrons, this orbital stabilization in a way is greater than this electrostatic one. Okay. Okay. So now, okay. So where was I? Okay. So now what we are going. So location is also, yes, I was there. Location is also very important to characterize to understand the bioactivity of a molecule, because organisms are with compartments or organs, but cells also, they have different compartments. And so this is why we develop metal complexes. We want to characterize their activity, and the first interest that we have is into antioxidants. But then I will show you that we try to develop methods for direct imaging at the subcellular level of metal-based system. So I will start with the catalytic metal-based antioxidant. And what we are doing, we are trying to, so we are using a bio-inspired approach, and we try to mimic the activity of superoxide dismutase. So superoxide dismutase are metalloproteins that are very important in the cell. They are protecting the cell against a reactive oxygen species, so a species that is derived from the oxygen. The oxygen not very reactive because of its triplet state, but if you reduce the oxygen, you obtain a molecule kinetically, thermodynamically less reactive, but more reactive. And this is what you see here spontaneously. Superoxide is not stable, and it would react spontaneously to make this mutation. So one superoxide will be oxidized into the oxygen, and the other one will be reduced into H2O2. And this can be, so the spontaneous dismutation is very fast, but not fast enough to maintain a low concentration a concentration low enough in a biological system. And so you have something that is, well, let's say nature has evolved metalloproteins that can accelerate this reaction. And so this is a redox catalysis. And what I show here is just a simplified mechanism. You have a metal ion that will oxidize superoxide into the oxygen, and the reduced form of the metal ion will reduce superoxide into H2O2, of course, in the presence of protons. So what is very important is to remember that biological system, they use the oxygen, and there is a natural flow in biological system of species that are derived from the oxygen, more reduced than the oxygens, like superoxide, H2O2, but much more reactive. But this is a natural process. And so oxidative stress or distress is not this production of reactive oxygen species. This is the fact, because some of them are very useful, but if this the reactive oxygen species are produced in a too high flow, if we disrupt the balance between the antioxidant defense that exists in the biological system and the production of this reactive oxygen species, you obtain what is called an oxidative distress. And this is a condition where you have oxidative damage in macro biomolecules like DNA, lipids, proteins, and these are irreversible, and they may lead to some pathological condition. And so for us, we have been interested into in this protein, which is MNSOD, and that we use as a model to try and design some small complexes. So I like this picture. It has been drawn by one of my former PhD students, so Émilie Mathieu. And in France, we have a contest that is called Speak About My Thesis in 3 minutes. And so you have one slide, and then you explain to historian, physicist, mathematician, what you do. And then there's a contest in each university, and then after there's a contest for all France. And so she participated to this contest, and for that she used this picture, and that represents exactly what we are doing. So we So we work in the chemistry lab to design the molecule, but at the same time we are putting this molecule directly in cells and trying to characterize their distribution. And mitochondria, this is what she shows here, is of a particular interest for us. So we'll come back. I will come back to that. So now we want to mimic the function of superoxide dismutase, and to do that, because there is we have a model that is the SOD. We will try to reproduce the active site, and this is what I have. I show you here with this picture that is from Magritte. So Magritte, so it's a self-portrait that you see here, and in this self-portrait, so this is Magritte that paints a bird looking at an egg. So looking at the egg is able to think about what the bird will become, and this is why it's called la clairvoyance, which means that he can foresee what this egg will become. So I have removed the bird and the egg, and I have put at the place of the egg. You see that there is the protein, and what is important as a chemist is really the structure of the active site. So this is what this allegory, in a way, of bioinorganic chemistry means. Okay, so what are the important parameters of SOD, and what are the physicochemical parameters that are responsible for this very interesting antioxidant or anti-superoxide activity? So what is interesting is that you have several proteins that are able to do this function here, several metalloproteins, and they can have different metal ions at their active sites. So we can find iron, manganese, copper, or nickel superoxide dismutase. And if you're a chemist, you know that all of these metal ions, they show very, very different redox properties. I've shown here the redox potential of manganese 2 in water, manganese 2, manganese 3, and the redox potential of iron 2, iron 3 here also in water. So you see, they are very, very different. And what is impressive is that if you look in the literature and find the redox potential of the protein, all of the SOD that have been published, copper, manganese, iron, nickel, it's a little bit more complicated, but let's say just copper, manganese, iron, and copper, iron, and manganese. They all fall in a very narrow range, and that is indicated here in blue. And what is very interesting is that this narrow range does correspond to the optimal redox potential for the superoxide dismutase activity. And what is this optimal redox potential? This is the midpoint between the redox potential of the couple where the superoxide is the oxidant, and the redox potential of the couple where the superoxide is the reducer. And this taken, of course, at pH 7 because they are pH dependent. So we have to take into account that. But then if you take into account the pH, you obtain this optimal value. And then that shows us that the role of the protein around the metal ion is really to control the redox potential of these metal ions. So this is what nature does. And so this is an indication for us as chemists that this is what we have to aim at. So we have to try and reproduce this redox potential. So to do that, and this is what I was doing in Orsay, we have developed a range of different ligands that reproduce maybe I can show you again the active site. So if you look at the active site here, you have a trigonal bipyramid structure with 2 histidine in a triangle and 1 carboxylator. And in the apical position, you have 1 histidine that is a bit farther away than those in the plane, and a water molecule that can be water or OH minus. But this triangle is very important because superoxide arrives in between the 2 histidine that you see here. We know that because people have studied an inhibitor of these proteins, which is azide, which resembles superoxide but cannot be reduced, and it goes exactly in between these two amino acids. So first we thought, okay, let's try to have a system with a low density, and that would reproduce this triangle and this apical direction. And then we vary systematically the nature. So the first one, which is called BIG, reproduces 2 imidazole and 1 carboxylator to reproduce approximately what is at the active site of the protein. And then we have changed systematically different things. So you see that we have changed imidazole for a pyridine, for instance, here. Then we have introduced pyridine with bulkiness. Then we have changed the carboxylator with a phenolator. And doing that, we were systematically looking at the redox potential and the superoxide dismutase activity. And what we obtained, this is the trend that you see here, the one that is in black. I will discuss the pink one later, but the one that is in black here. And we see that by the redox potential is above the optimal redox potential. And if we reduce the redox potential, we increase the kinetic activity. So that makes completely sense. And what is interesting is that there's another family of molecules that is completely unrelated on the structure, which are porphyrin that have been developed by a group in Duke University, Ines Batinic-Aberle. And for this porphyrin, so I've been also interested in porphyrin, so this is why I looked at that. They are stable at the manganese 3 state, whereas we are stable, these molecules are stable at the manganese 2 state. And in contrast, to favor the activity here, we need to increase the redox potential. And we have like a mountain plot, and you see that these 2 unrelated family, they do cross approximately at the optimal value for SOD. So this is really to say that the strategy was the correct one, and this redox potential is very, very important for this activity. So now we end up with a series of molecules, one that is about 1% of the activity of the enzyme. But if you remember, I didn't stress that, but the enzyme, the activity of the enzyme, so the natural dismutation of superoxide is of the order of about 6×10 to the 5. And when it is catalyzed by the enzyme, it's 10 to the 9, which means that the activity of the protein is only limited by diffusion. So these proteins are very, very efficient. So here we have a molecule that is about 1% of the activity of the enzyme, but that remains good because the enzyme is just limited by diffusion. So now that we have Oh, the porphyrin, what they have tuned. So you can, it's in the porphyrin system, it's very easy to tune the redox potential by modifying both the pyridine that are present here and the X that are the substituent on this pyrrole ring. That's very, very easy to modify that. And what they showed is that the the positive charge here is very important to make an attraction of superoxide, so that in addition to the redox potential, there's also an effect of the charge. And with a similar redox potential, if you have or not the charge, you have a different kinetics. So they both modulated the redox potential through this substituent here on the pyrrole ring, and the charge through the substituent on the pyridines that are perpendicular to the porphyrin ring. Does that answer your question?
Yeah, yeah. And the actual ligands, I mean, water?
No, they were not playing most with the actual ligand. No, that was really.
So that is water or something?
I'm not sure that in the manganese 3 state, maybe there's a chloride that stays on the porphyrin, but I'm not fully sure here. So I know that they isolate it with chloride, but I don't know if it stays as an actual ligand of it's a manganese 3, so probably there's no, because then it's oriented in effect, so it's probably without any ligand. Probably. I'm not sure, but I can look if you're interested, and I can tell you. Okay.
Thanks.
So there are other parameters that I'm not going to discuss today, but if you want, we may discuss after the talk. So there's also a charge effect that I've just mentioned, and the question of the compartmentalization of SOD, the SOD that I've mentioned. They are very large, negatively charged protein, and they stay in the compartment where they are produced. So it could be where they are at least produced as a metalloprotein. So if it's in the mitochondria, it stays in the mitochondria. If it's in the cytosol, it stays in the cytosol. So there's some kind of a compartmentalization of SOD, and we think that the targeting to some specific organelles can be very important, but I am not going to discuss that much today. So we developed different kinds of modification. First, we were interested in peptides, and we functionalized this kind of molecule with peptide, and this is mainly for targeting, but again, I'm not going to discuss that today. Nicolas Delsuc in the group is also developing a design that is combinatorial design of peptides. So here, the peptide is not used as here for targeting, but for direct coordination of the metal ions. And then by screening with different activity tests, SOD or CAT, we're able to generate, so not manganese, but copper complexes that were mimicking either SOD or catalase activity. And then I just added this transparency because I've discussed with some of you this week, and I know that you use this kind of fold of protein that you characterize, and we also develop, we use this kind of fold and we mutate. So this is larger, I would say, or more extended than this just oligo amino acid here. And this fold, it consists of 3 helices that interact with each other. And by mutating amino acid at the center here, it's possible to engineer an active site, a site that is able to coordinate metal ions, and if you choose correctly the nature of the amino acid, then you can reproduce the correct redox potential and obtain a mini protein like that that is active. But I'm not going to discuss about that. So now, what I'm interested in as an inorganic chemist is I have these molecules. I know they're active against SOD. Well, I know, well, no, not yet, because I've not discussed that, but they have been used to reproduce the active site of superoxide dismutate. How can we evaluate that? First, we need to evaluate them out of any cellular context, and we need to determine the catalytic constant for reaction with superoxide. For that, we use several techniques. It can be stop flow or pulse radiolysis, and this enables us to put our complex with a in front of, let's say, a large excess of superoxide, and so we can know if the system is catalytic or just stoichiometric. Then we can also go a little bit more towards a biological system and use a slow flow, because this is really what happens in biological system. There's not a burst of superoxide, so except in some very specific occasion, but normally there's just a slow flow of superoxide. And there's an essay has been designed by Fridovich to make this characterization of the activity with this slow flow.
So
And
just-
then,
can I ask a
yes,
question,
yes.
just stopping you for a second?
Yeah, no problem.
So you have these plethora of complexes that you have made, the inorganic complexes, right?
Yeah.
And they are all of different design, whatever the design, it could be helical, in the helix, sorry, or it could be peptide based or organic based. They all have different solvents, right? They go into, right?
No, they are all soluble in water. We make
all of them.
All of them. So the inorganic, the one that are the most artificial non-peptides one, it depends of the counter ion. So if you isolate with PF6-, for instance, then you can crystallize, but you cannot solubilize. And we change the counter ion, and then they are soluble. So now we all use them in a soluble form.
In water.
In water, yeah.
So everything is in water, and then you put the superoxide. You purge it with, okay.
So we can also characterize the activity in anhydrous medium like DMSO acetonitrile. So this is possible because we can have superoxide in this context. So this is also something we do, but they are also soluble in acetonitrile. So yeah. Okay, so this enables us to characterize what I would call the intrinsic activity. That is the activity of this molecule. Does it react fast with superoxide when there is just, I would say, in the solution, the solvent, the complex, and superoxide. But of course, in cells, it's much more complicated. So the question that arose is, does that reflect the activity in a biological context? So in fact, if you think about bioactivity, it's not only associated to the intrinsic activity of the molecule, but also to parameters, as I've already said, such as location, speciation, and quantity in cells or at the target, maybe for us, the mitochondria. So there's a requirement for the characterization of the bioactivity, and we need to go from the to take these inorganic complexes and try to put them in cells and to ask us the questions: how efficient are they in cells? Where they are going? What is the nature of the active species, and how many of these active species are they present in cells? And this is what I call inorganic. Chemical. Yes, I will. Inorganic chemical biology. And again, we go from the round flask to cells, and this is what I'm going to tell you about now. So so as I said, we met with these medical doctors, and it happens that they had in hand a cellular model that are epithelial cells from the gut, intestinal epithelial cells, so HT29, and they have modified these cells to overexpress a soluble protein that is MD2, and MD2 is a protein which is a co-receptor of lipopolysaccharide, bacterial lipopolysaccharide, and that makes these epithelial cells from the gut, intestinal epithelial cells, very, very sensitive to lipopolysaccharide, and they would, in the presence of this lipopolysaccharide, induce an overproduction of inflammatory marker, And so this is the way that we do the assay. So we take, we grow cells in a plate. We make some incubation with the complex of interest. We activate the cells. And then we take the supernatant in which we measure the inflammatory marker and also the leakage of LDH. And inside cell lysate, what we measure are other inflammatory marker like COX-2, and also because the idea is that this LPS-induced inflammation was said in the literature to be associated with oxidative stress. So we decided also to characterize the modification, targeted investigation of the modification of proteins like SOD and catalase, and to measure also the toxicity we need to measure. So we do that for each well. Here we measure the LDH in the supernatant and in intracellular, in the cell lysate, and we have an information of the toxicity for each condition. This is something important because if we have a cell that is in bad shape, it will not produce as much as EL8 or as much modification of SOD and so on. And in fact, this would be a false positive result. or false negative, depending on what you look at. But anyway, okay. So now we have this assay, and we want to characterize this manganese antioxidant. So this one, MN1, the one that is this 1% activity in comparison with the protein. So the first type of characterization is quantification, and for that, we can use for manganese a simple way. We just use the cell lysate, we acidify the cell lysate, and then we can we obtain a very, very nice EPO signal. So never mind why there is 6 lines, but it's connected to the nuclear spin of manganese, and we can quantify that. If you have a metal ion, for example, sometimes we use zinc that is not EPR active, then we can use ICP-MS like indicated here. And we have compared for manganese EPR and ICP-MS, and we obtain the same quantification information. So this is the total of manganese that is inside cells that we have added, and we can suppress, of course, the endogenous manganese, but we have an information of the total manganese. But it's possible that just this complex is just a manganese carrier. It has been suggested by one referee at some point when we were submitted an article, and so we wondered, okay, we do not want only to measure manganese, but we want also to characterize the speciation. That means, do we still have in cells this molecule, or is there another molecule that is present? And to do that, we can perform a speciation. So depending on the stability of the complex, we can use mass spectrometry directly of the cell lysate. Or another technique, if you're interested, I can discuss with you, but this is the reference is here in blue. We can use ion mobility also to make this characterization for the system that are the most labeled one. Then we want to characterize the imaging. Okay. Oh, okay, never mind. We want to characterize the distribution of the of manganese in cells, and to do that, we do not want to modify. So we could have taken this molecule and put a fluorescent probe, but a fluorescent probe is always a very large hydrophobic molecule, so that would have changed the physicochemical properties of this molecule. So we said no, we want to just do imaging of this unlabeled molecule, and we decided to go towards X-fluorescence and imaging manganese. So of course we have compared the image that I'm showing you with endogenous manganese. No, there's much less endogenous manganese than what I show you here. So X-fluorescence is a very interesting technique because it can image any heavy element, any element heavier than silicium. Okay. So potassium, for instance, is known to be homogeneously distributed in the cells. It can be imaged. And then you see here a map of potassium homogeneously distributed in the cell. And what we see in X-fluorescence is when the beam, in fact, crosses it's not confocal, this is what I mean here, and it shows you the amount of potassium in the section of the cell. And so here we see that this element potassium that is homogeneously distributed in the cell, there's a higher quantity at the center of this image, just because this is where the cell is thicker. And so you have with potassium the distribution of the shape, of the cell. And we see that in this case, so with this molecule, the manganese is distributed approximately homogeneously within the cell. Okay, so these are the physicochemicals.
Fixed cell or light cell?
No, no, you cannot do lab-state in X-fluorescence because you irradiate with a beam that is very, very energetic. This is about, I think here it was at 8 kilo electron volts, so you would generate a radical and so on, and we need to do that for several hours. Okay, this is not that responsive. So we do that depends on what we want to do. If we have here a molecule that is diffusible, we need to perform cryofixation, lyophilization to perform this kind of imaging. Yeah. Characterization. Now, what about the bioactivity? So now this is what I show you. So I just show you here EL8, but we have a similar profile with COX-2. And what you see here is that With the control cell, you have a very little EL8 production because there's no inflammation. When we add LPS, there's a huge increase, and this is exactly what we wanted to have. We wanted to have something with a large increase of an observable parameter, here the quantity of EL8, so that we could see the return back to normal in the presence of our molecule of any antioxidant, by the way. And what is important here is to remember that in bioinorganic chemistry, there's a lot of, or medicinal inorganic chemistry, there's a lot of metal complexes that are used. And at the moment, they are, if you think about what they are used for therapeutic application, it's always for a toxicity. And in fact, a medicine is very frequently a toxic molecule, but it is with a selective toxicity. So it can be toxic for bacteria, an antibacterial agent, and not for eukaryotic cells. It can be toxic for cancerous cells and not for normal cells, so for anticancerous agents, and so on. And so most of the cellular assays that you have in the literature are based on toxicity measurement. And here, as I've explained to you, our project starts in a way when the molecule is not toxic, because what we need to have here is a molecule that does restore the normal functioning of a cell under some kind of a perturbation, here oxidative stress. And also, this is important to realize that because we tried, so here, of course, I show you the results, but it was not the first model that we used. And it is sometimes very difficult to perturb a cellular system so that you can see a modification of a specific parameter, but not too much so that it's possible, so that the cell do not die, because we do not want them to die, and that in the presence of our of this agent or of a therapeutic agent, we go back to normal. So you see there's some kind of a compromise here between we want to induce a huge effect, but we do not want the cell to die. And that was the difficult thing to find. And with this cell, we have something that works very well. Yeah. Yeah, no, there's no silly question, so go on.
Yeah, isn't there already SOD in these cells actually?
So I'm going to that. Can I go on? I'm going to that. So yes, because, okay, now we have that, and we wondered, is that associated with the antioxidant activity? So we tried some superoxide markers, and here in our hands, they were not very, so we were able to manipulate them, and we saw that the manganese complex reduces superoxide, but we never saw this increase that I'm showing you here. And we think this is very important to see that increase. And we think that this is because there is some kind of a cellular response to superoxide, and the cell is able to buffer superoxide because it has SODs. And this is what I'm going to show you now. And so we have also measured, so in our cells, in the human cells, you have three different SODs. So one that is MNSOD, which is in the mitochondria. One that is a copper SOD that is in the cytosol, and there is also one in the nucleus. But here what we've measured is the copper SOD in the cytosol, as you can see here, so against actin. And the copper SOD does not change with the different conditions. So the conditions are controlled, LPS plus MN1. I will go back to that later, okay? And so we see that the copper SOD does not change. Now, if we look at MNSOD, upon LPS activation, we have so it's after 6 hours, we have a huge increase in, well, huge, measurable, well, huge, but measurable, let's say, an increase of the expression of MNSOD, and we have checked that it is in an active form. We have also an activity, a gel activity. And what is interesting is that in the presence of MN1, that means if we co-incubate LPS with MN1, this overexpression is reduced. And we think that this is an indirect proof that the complex is actually complementing 4-MNSOD. That means that the cells that needed in the presence of LPS to overexpress superoxide dismutase MNSOD because of this oxidative stress, so the reaction of the cell is to protect itself against oxidative stress, this is no longer needed in the presence of MN1. So that shows that MN1 has this activity in cells. And what is interesting is that it is in the mitochondria and probably not in the cytosol. So this is why we think that it is important to target the mitochondria. And now we have some negative control. We test MnCl2 because this activity, it's associated with MN1, but MN1 can release its manganese, and we see when we test MnCl2 that there is no activity of MnCl2. And then we wanted to test the ligand, because it could be also associated with the ligand. The problem is that the ligand itself is very toxic. So in the system that I've presented you previously, we couldn't make any measurement, or we could have, but we would have something that would be a false positive results, because of the toxicity of this molecule. So then we turn to the measurement of the zinc complex activity that was also found to be inactive. And similarly, in this assay, zinc and MnCl2, zinc complex and MnCl2 have no activity. Okay, so this is the end of the first part, but maybe I will end here, and you will ask me all of the questions. Maybe I will show you one image, but I hope I have convinced you for this part, at least for this part of our work, that Mn-based catalytic anti-superoxide agents can be designed, and that as chemists we can develop systems, cellular systems in which we can study them for their bioactivity. But here we have really to design some specific systems, and I've shown you that it's very quickly that it's possible to determine their quantification and location. So now what are our applications for this part of our work? So we want, of course, to develop these catalytic antioxidants to limit oxidative stress in pathology. So I mentioned inflammatory bowel disease, but it can also be Alzheimer's, for instance. So we have a paper on that if you're interested. And we can also use that to limit oxidative stress-induced secondary effects of drugs like oxaliplatinum. Oxaliplatinum, you have a neuropathy that is developed when you are cured with oxaliplatinum, and this secondary effect is associated to oxidative stress. And so we've tried our molecule, and they work to limit the neuropathy induced by oxaliplatin in mice. We are not farther away, but in mice. And of course, and this is the reason I'm here, we want to study them with manganese probe, and this is why we collaborate here with Ancona Data. So maybe, so just one word about metal-based tag. I just want to show you, because I think it's something really beautiful, how we use, and this is different from what I have said until now, so that can maybe we can discuss about that after I finished. So just about these molecules. So these are interesting molecules because they have a modality that is associated with the presence of the CO that does correspond to a very huge absorption in the 2000 centimeters to the -1. And this is an area where approximately nothing absorbs in biology. So this is interesting, but they also show a fluorescence activity that is not a very good activity, but we can use that to validate the molecule. And also we have a heavy element, and because we have a heavy element, we can image that by X-fluorescence, as we have done, I've shown you for manganese. And we have developed this molecule with this ligand because they are very easy by just click chemistry. They are very easy to functionalize to any kind of substituent. So I'm just showing you two images. So this one and the next one. So infrared. So we can image. So normally, if you are a physicist, you know that we cannot perform infrared at a resolution that is, so if we speak about 2000 centimeters to the -1, that's 5 micron. Because of the diffraction and Haber criteria, it's not possible to have a resolution if you use optical detection. It's not possible to have a resolution that is better than 2.5 micrometer. But this is with an optical detection. And as a physicist, in Orsay, that has developed a very, very clever technique that consists of an AFM coupled with an infrared laser. And what it measures, it's infrared absorption through a transient dilatation of the cell upon heating, because when you have the absorption of an infrared wavelength by a molecule, it heats the environment. And so you have a transient dilatation because the laser is a pulse laser anyway. So what it measures is with an AFM tip, it measures the oscillation of the AFM tip. And we've shown that with this kind of molecules that are used for, that are anti-cancerous agents, so without any label, we were able to map their distribution. So this is what you see here. So we have hotspots of this rhenium 3CO core, and they do correspond these hotspots with the nucleus. So this is the first example. And now, what about? So I just don't show anything about the design, but what is interesting in excflorescence? So of course, we need fixed cells. We cannot do movies. So it's not as fancy, I would say, as the classical fluorescence, but it can give you some very interesting information, especially if you work with metal ions. And so if you want to image, so now if you compare with a classical fluorescence, now there's a lot, you can buy any marker. If you want to check that the molecule is at the mitochondria, you buy in vitrogen a marker of the mitochondria. There's no such thing in excflorescence. And so because we needed that, because some of our complex accumulate in some compartments, we wanted to know if it was the mitochondria or not. So for the nucleus, zinc accumulates at the nucleus. So you have an endogenous marker of the nucleus, but for mitochondria, this is not the case. And so we have developed these for our purpose, but of course, now it's a molecule that is available. This rhenium 3CO molecule with a trisphenylphosphonium. And trisphenylphosphonium is a very lipophilic positive charge molecule. And so it accumulates at the mitochondria. And this is what you see here. And we have shown that in classical fluorescence. This molecule goes at the mitochondria. And you can observe it by excflorescence. And you see a perinuclear localization. So now if you have a molecule like our manganese complex, now we're able to co-image this kind of molecule with a manganese complex just with one experiment. We don't need to do a lot of fancy fluorescence and recover the same cell in excflorescence. Okay, so just to show you that also as an inorganic chemist, we also develop inorganic tools for our purpose, but for anyone's purpose. Okay, and so just this was the second part that was very, very fast. I just want to thank the funding and particularly CEFIPRA because, because I'm here thanks to, it doesn't work. Okay, anyway, I'm here thanks to the CEFIPRA project. So if you're interested, if your students are interested, so we are now recruiting for a postdoctoral position for delivery of sun mimics. And also a PhD position for the development of sun mimics. These are the people that we have been that have done that work. So Hélène Bertrand, Nicolas Delsuc, and PhD students. So particularly what have been discussed. Okay, it doesn't work. I've been discussed the work by Anne-Sophie Bernard and Émilie Mathieu. And the work of he's not here. I forgot to put him. Well, the work of Sarah Ostachi and Gabriel Chan, who's here for the development of. Sorry, I am not used to this point. Okay, well, anyway, Gabriel Chan, who's here for the development of a system that can be used for tracking mitochondria. Okay, just a few words about where I come from. So I come from France, and my university is in central Paris, very close to maybe a famous building that you know that is called Le Panthéon here. So I'm the orange circle, and the Panthéon is in yellow. And these are some pictures of the school. And so it's an historical building also because Louis Pasteur, you know, he was a chemist. He was not a biologist. Then he did biology, but he was a chemist. Yes. And so he did all of his work, most of his work, I would say, at So if you come one day, I can show you his office that is still there. So here are some pictures of the students. And I just wanted to say, but I've already discussed that with Yodin, that we have some international fellowship to study at ENS. And so you can have a 3-year fellowship to study for 3 years at the master level. Okay, and I'm also the president of SBIC. So if you're interested in bioinorganic chemistry, please join SBIC. It's very important well, yes, to be all together in a same society. And thank you for your kind attention. Sorry if I have been a bit long.
Thank you very much, Dr. Thank you very much. We have questions. Time for questions. Okay, yeah.
In one of your slides about bioactivity, you mentioned speciation. Were you talking about speciation in the sense biologists talk about, like something evolves into a different species?
So I was speaking about the fact that this manganese complex can dissociate in cells
Yeah.
And so there's so we were not looking at what the manganese was becoming, but rather about the ligand that was present in a rather high concentration, what metal ions goes inside the ligand. And we see that and this is why we tested also the zinc complex. So this is the speciation of the complex in a way. So when manganese goes away, there's zinc that goes in, and this is this kind of speciation that we look at. So I could have said also that we check the integrity of the complex, but with this technique I've mentioned with mass and ion mobility, you also have the information of the older complex that are formed, like the zinc one, for instance, and which can be, depending on the nature of the complex, at a very high proportion. So this is why I'm speaking about speciation, because we'll not only look at this molecular species, but also at what the ligand becomes. So it's a little bit different from speciation that geochemists use, because geochemists, they would have looked at the manganese and what is the form of manganese, but we know that this one is not active. So I was really looking at what is happening to the ligand.
Yeah, I totally just following up my question about SOD. So you mentioned that your molecule actually acts complementary to SOD that's present. So I'm wondering, in the cell expression levels for SOD, right? That would be responsive.
Yeah.
If there is some other agent, for example, which is present, right? So the expression level should go up and down. So I don't
what happens is that when you have oxidative stress and when you have the production of superoxide, there are some sensors, and then the cell will overproduce MnSOD. Well, in that case, MnSOD only, not copper. So this is why we think that the superoxide is mainly present at the mitochondria, and for some reason, there's a lot of respiratory complex and so on. So this is probably why it is at the mitochondria that can be decoupled and so on. And so what we see is that when we put the manganese, there's a diminution of this overexpression. So this is why we think that superoxide takes care of this complex takes care of the superoxide at the very beginning of the system. Okay.
Yeah.
And so it reduces inflammation because and now we've done some kinetics to try and grasp this idea that it goes at the very beginning. What is interesting is that, so I didn't mention that, but the porphyrin, they are supposed to induce an antioxidant effect, but differently from our complex, because this porphyrin, because when you dismute superoxide, you produce H2O2, and if you do that, so our complex is good, but not as good as superoxide dismutase. So that means that the H2O2 that is produced at a slow flow by our complex. Whereas with the porphyrins, they are much more effective. The catalytic constant is much higher than what we have. Some of them are very close to the activity of the SOD. And then what they see, so we are now trying to look at that also, but what they see is that there is when they put the porphyrin, due to the presence of the porphyrin in oxidative stress context, there's an overexpression of SOD. So it's a different pattern from what we have. We have a decrease and they have an overexpression. And when you have this overexpression, you have a long-lasting antioxidant effect, okay, because you produce this protein. And so probably the antioxidant effect of this protein, of this porphyrin, paradoxically is associated with a pro-oxidant effect, so that they induce a burst of H2O2 that activates the what is called the Auro system, so antioxidant response element of the cell that produces SOD. And in fact, probably the anti-inflammatory effect or the effect of these porphyrins could be associated, in contrast to what we have, not to their direct removal of superoxide, but to the fact that they overexpress SOD. So a cocktail of this system could be interesting at some point.
Okay, I have this question, since there aren't questions that I can see. I have this couple of questions. One is, of course, you have synthesized all these complexes, different kinds of things, and are delivering to the cells. Do you know what is the time scale of the uptake? And also time scale of the uptake?
Okay, yeah,
and the second thing is, what happens to now the superoxide sensing ability within the cell? So SOD is expressed when it senses genetically, right? That you have to express it up. So do these metal complexes actually now divert from the sensing ability of the cell?
Okay,
Because-
so
Because that is a complex.
very good question. So the first part, so we've studied that with one of the complex, and we see that so they enter rather fast, but there's a so the leveling is at about 2 to 3 hours.
Right.
And so it increases by a factor of 3 to 4 the amount of manganese in cells. And if we just put MnCl2, this is about 7 times at the same concentration.
Right.
Okay, so it's at the order of the hour, but it starts to come in at the very beginning, but there's a slow increase
Okay.
up to 2 to 3 hours.
Okay.
And so what happens? So no, so I've not looked at that. What happens at the sensing? Because I don't even know. I have to look into that, but I don't even know how the cell is sensing superoxide. You have some idea?
Yeah. No, this would be
We
interesting.
have not looked
too.
into that. What
Yeah.
we have looked now, it's not published, but we are trying to publish that. So now here I said that we do targeting. We look at we do targeted, not proteomics, but we look at the proteome with a targeted aspect, as means we look at thought, catalase, and so on. And then we said, okay, can we do that without any targeting?
Sure.
Okay, and so yes, we can do proteomics.
Sure.
And what is very interesting is that what we see is that when we put LPS, we have overexpression and underexpression of a range of systems.
Lots of things.
And when you put manganese 1, everything goes back to normal, with the exception of one protein that is associated to wild toxicity, but most of them go back to normal. So I think this is showing that, so that I think this is really, that does correspond to the fact that this complex acts at the very onset of the oxidative stress.
And it could be related to sensing,
It could be
maybe.
sensing. It
Yeah,
could be
yeah,
sensing.
yeah.
So yeah,
That's very interesting.
Good question.
Okay. Okay. If there are no more questions, let's thank Claudine once again for a very nice talk, and we will all walk with you to have some refreshments outside the West Canteen. So thank you.
Thank you very much.
Thank you.